I was thinking I should say something in Spanish to start with, so I'm just going to say buenos tardes a todos. And then, preparing this very short, easy sentence, I realized that, so I'm, as Maxi said, I'm from Belgium, from the French-speaking part of Belgium, not Brussels. So all this area is called Wallonia. And in Wallonia, there is a dialect, which is Walloon. And in Walloon, I would say not buenas tardes, but just bonjour. But a todos is a tortos. It sounds really similar. I never realized that before. So you see, Latin language probably. So let's be a bit serious. First of all, I would really like to acknowledge Maxi and Pepe for organizing this summer course, and even more importantly, for having me here for the third time indeed, at this particularly nice period of the year. You know, Belgium is not very famous for its weather and climate, but especially in January, I can tell you this is really, really uncomfortable and unpleasant. I'm still wondering how this guy stayed for such a long period. But okay. So as Maxi said, in my scientific career, I've been doing mainly enzymology and protein folding. And recently, we changed gear a little bit. And I'm going to present you a rather recent project of the lab, which is related to the study of bone morphogenetic protein, which I'm going to call BMP, and in particular with BMP2 and the antagonist Noggin. So, as you can see on this very simple, schematic, and not so recent phylogenetic tree, these BMPs, together with the growth and differentiating factor here, form the largest group of the TGF-beta, the transforming growth factor superfamily. And these BMPs are multifunctional cytokines. So cytokines are simply secreted cell signaling proteins. Multifunctional cytokines that act in a highly context-dependent manner. So they have plenty of different signaling activities. But they were discovered, as you can see, this is the reference, they were discovered in the mid-60s by an American surgeon called Marshall Urist, who found out that these proteins trigger the formation of bone and cartilage from stem cells in culture. And this is why they were called bone morphogenetic proteins. But as I say, they have plenty of other activities. So these are very versatile growth factors, which, as you can read, they act as morphogens in embryonal development. They steer organ formation. They mediate tissue homeostasis and regeneration in adult organisms. So they are involved in the maintenance, in the differentiation, in the proliferation, in the regeneration, and also in the death of cells. So really a lot of various activities. Because of their involvement in the maintenance and repair processes of various tissues and organs, these BMPs show or display highly interesting, or constitute highly interesting targets for novel pharmaceutical application in regenerative medicine, and in particular, in cartilage and bone regeneration. And in particular, BMP2 and BMP7 were approved by the FDA to be used as possible drugs. It was also demonstrated and shown that the deregulation of the BMP signaling pathways is associated with a whole series of diseases, for instance, dysfunction of the osteoarticular, central nervous, and cardiovascular systems, and also, guess what, with cancers. And so they show really a clinical potential in various diseases, such as musculoskeletal disorders, vascular syndrome, and cancers. So for this study, we decided to focus our attention on BMP2, the X-ray structure being schematically represented on this picture. So this was solved, as you can see, a bit more than 20 years ago. All these molecules are dimers. This is really obvious here from the color code. So one monomer is in light blue, the other one in dark blue. And these two monomers are covalently linked here by an interchain disulfide bridges. Now, each monomer consists in a series, in this structure there are 8, it doesn't really matter, 8 beta strands, which forms a pair of antiparallel beta sheet. And within each monomer, there are 3 additional disulfide bridges, which forms a very distinct motif of all these TGF-beta factors, known as a cysteine knot. If we zoom on it, so you see the 3 intrachain disulfide bridges which form the knot. 2 of them forms a ring, whereas the third disulfide bridge really protrudes, really crosses the bridge. And so, as you've probably seen already, this protein is rather small, with a molecular mass of about 26 kg. This picture shows you a very simple, very simplified, but this is much more than enough for us at this level. It shows you the mechanism of BMP signaling. So 19 BMPs have been characterized in human, and they all signal the same way. You see a dimeric BMP binds to a dimer of type 1 and a dimer of type 2 membrane receptors, and these receptors display serotonin kinase activity. And the binding of the BMP to these 2 monomers of BMP induces the formation or trigger the formation of an heterohexameric complex, and this triggers a phosphorylation cascade. So the type 1 receptor, which is a constitutive kinase, phosphorylate the type 1 receptors, and then this one, type 1, induces or catalyzes actually the phosphorylation of 3 different receptor-regulated SMET proteins, term 1, 5, and 8. This phosphorylation triggers the association with SMET4, so 3 different kind of heterodimers formed, which can then translocate into the nucleus. And these heterodimers triggers on their own, or possibly, but not possibly, this has been shown, but this is not completely clear yet. They trigger, so on their own or with some transcriptional partners, they trigger the transcription of some target genes. This mechanism is highly regulated, both outside and inside the cell, and also in the membrane. So outside the cells, a number of molecules that are known as antagonists have been described. In this work, I'm going to tell you about Noggin. We also have worked a bit with gremlin, but I'm not going to detail this. So the binding of these various antagonists to the BMP simply inhibit binding to the membrane receptors. In the membrane, there are, as you can see, stimulatory and inhibitory co-receptors, which, without going into any details, which actually I don't really know, which promote or inhibit the signaling by the BMP. And finally, inside the cells, there are also a series of inhibitory constraints. For instance, there are other SMET proteins which interfere with the association of these 2. There are microRNAs that also play a role. And not on the picture, I know there are some phosphatases which are simply going to hydrolyze and remove the phosphoric groups, and so inhibit the signalization. And finally, to be absolutely complete, hola, the best journalist I've ever met, after my wife, actually, because she's a journalist as well. So this BMP also activates MET-independent signaling. Don't feel annoyed, Fabiola. They all know how good you are, otherwise you wouldn't be there. Okay, so on the top of the structure that I've also quickly described, the binding sites to the 2 receptors have been highlighted. And actually, well, this is something I quite like. The structure of the BMP has been compared to 2 hands positioned like this, okay, 2 monomers. And the wrist, this part of the body, or the hand actually, corresponds to the epitope binding to the type 1 receptor. then the palm corresponds to the cysteine knot, and this part, which is called knuckle in English, is the binding site to the epitope 2. So this is just summarized here. So epitope 1, type 1, and epitope 2, type 2. It doesn't really matter because I'm not going to detail this further. Okay, I take it as a chance to have a short break. Sorry. So to finish with this introduction, as I told you, BMP2 has been really clearly demonstrated to be involved in both cartilage and bone formation through a mechanism that is known as chondrogenesis. So BMP2, so chondrogenesis consists of cell differentiation, so the stem cells differentiate into mature chondrocytes, and this is stimulated by BMP2. These mature chondrocytes are associated with the formation of cartilage, and then these mature chondrocytes can further differentiate, still promoted by BMP2, to form hypertrophic chondrocytes, which is associated with bone formation. Noggin, the antagonist that I mentioned at the beginning, is blocking the differentiation at this stage. Noggin is here, so it just binds to BMP and actually hide the binding sites to the type 1 and type 2 receptors. This is a schematic representation of the complex that Noggin forms with another BMP, BMP7. We discussed, so BMP7 is really looking similar to BMP2. These proteins are homologous. Now it's clear to all of you, the students, what homology means, okay? So you see, indeed, they look very much the same without going into any details. And in doing so, simply Noggin masks the knuckle and wrist receptors. So in the context, and this is the context in which this study was initiated, in the context of cartilage and bone regeneration, it would be important or interesting or relevant to engineer BMP2 with a decreased affinity towards Noggin in order to use BMP2 in regenerative medicine and without having the antagonist inhibitory effect of Noggin. So the objectives of this work were and are still to identify and characterize the residues of BMP2 that mediate the interaction with the antagonist, and to modify these residues using a protein engineering, namely site-directed mutagenesis, protein engineering approach, in order to modulate the interaction profile of BMP2 and to promote cartilage and bone formation. So for this purpose, we had first to produce the two proteins. So both BMP2 and Noggin were overproduced in E. coli. This is actually described in the literature. And so we knew this would be under the form of inclusion bodies. Then these inclusion bodies were solubilized in a very classical way with urea. Student, you know that urea is a chemical denaturant, and DTT, a very classical reducing agent. Okay? This was described by Pepe on the first day. Then the two proteins solubilized under these conditions were simply diluted in a so-called refolding buffer to refold the protein, and this could be achieved in a single step. Then the two proteins were purified. In the case of BMP2, we use hydrophobic interaction chromatography because this protein is known for being quite hydrophobic, and Noggin was purified by very classical cation exchange chromatography. And finally, and this is also why I decided to present this data, we applied the quality control test that we discussed this morning. So the quality of the final samples, both BMP2 and Noggin, was assessed according to the list of recommendation tests, so the QC guidelines, established by these two networks, Arbre-Maubieu and P4EU, that I explained this morning. So these quality controls are all summarized on this slide. The data for BMP2 are at the top, on the top of the slide, and the data for Noggin are at the bottom. Classical, boring, but really essential. SDS page for both proteins indicate, well, let me go into a bit of the details. In both cases, the central lane corresponds to the molecular ladder, okay, that's the reference. On the left-hand side, in both cases, the protein was run under reducing conditions, so SDS page, SDS, plus DTT to reduce the disulfide bridges. And on the right-hand side, in both cases, we use non-reducing conditions. So remember, BMP2, there are 7 disulfide bridges, and I haven't shown that yet, but within Noggin, there are 9. So under reducing conditions, you see that both proteins runs to the gel and display an apparent molecular mass which corresponds indeed to what you expect in both cases for the monomer. Also not shown on this picture, but we used a specific antibody both for BMP2 and for Noggin. This is called a Western blot to show that these bands indeed corresponds to BMP2 and Noggin. And now under non-reducing conditions, you see that BMP2 migrates to an apparent molecular mass of about 25 kD, which corresponds with what we expected, indicating that this protein was indeed purified as a monomer. In the case of Noggin, this is a little bit less clear because you see there is a really important so-called smear effect, which, we have no explanation for that. This was, previously observed by other people under the same conditions, and okay, that's it. This is how it is. But I will show you with the other quality control assays that the protein seems to be fine. This was discussed several times during this week, of course. We perform a very simple UV spectrum. We measured the UV spectrum of both protein, absorbance versus wavelengths around 280 nanometer. I have to apologize. You will see that on all the spectra I'm going to show you, wavelength has been misspelled. We corrected that, but I forgot to change it. Never mind. So let me just remind you that this is very useful, first, to measure the absorbance and then the protein concentration at 280 nanometer, if you know the molar absorbance, of course. The shape of the spectrum, namely the ratio of the absorbance measured at 280 and 260, tells you that there is no significant contamination by nucleic acid. And finally, last but not least, no signal above 310 nanometer, as Pepe, I, and Human said yesterday. This is a good indication that there are no very large aggregated species in the solution, at least not in significant quantity. Finally, the first application of dynamic light scattering that Human mentioned yesterday, size exclusion chromatography combined with a multi-angle light scattering detector shows you that both proteins elute as a single peak. Okay, if you are a bit critical, and you should be, this one is not really symmetrical. You see there is sort of a tail here. Clearly, Noggin interacted slightly with the column, but nevertheless, there is just one peak, and the molecular mass in both cases, all across the peak, is unique or homogeneous, and you see that the calculated molecular masses are close to what you expect, right? And there is another parameter, which is not so interesting, but this is called the polydispersity index, I think, which is equal to 1 in both cases, indicating that both proteins are homogeneous in terms of size distribution. And finally, last but not least, this is really, as I said this morning, and I insisted, a very important quality control assay, high resolution, okay, mass spectrometry indicated that both proteins display the expected mass. So it means we are going to work really with the proteins with having the expected properties. And finally, okay, so this established purity, homogeneity, integrity, and identity, which is what we discussed this morning. And finally, to be absolutely complete, just for your information, you see that with BMP2, we could produce purified about 100 mg of protein from 1 liter of cell culture, less with Noggin, but this is still quite decent, 15 mg per liter. Finally, and this has been discussed several times during the week as well, optical spectroscopies constitute a very good tool for, let's call it, extended quality control quality. At the top, you have the fluorescence emission spectra of both BMP2 and Noggin. Remember from the lectures, tryptophan in solution has a maximum emission wavelength at 355 nanometer. In a folded protein, and yeah, fluorescence of proteins is essentially due to tryptophan. The other residues doesn't contribute that much, or actually not directly. So in a folded protein, the maximum emission wavelength is shifted towards lower wavelengths. In other words, it is blue shifted, and you see here there is a blue shift of about 20 nanometers, 12 here. So this indicates convincingly that both proteins display stable tertiary contacts. Now, far UV CD spectroscopy. So remember, we measure the ellipticity, we normalize the data. So these are molar ellipticities, and calculated MRW mean per residue. Okay, the data were normalized properly. You see that both spectra display rather intense negative signals, indicating that both proteins display some secondary structural organization. Here I'm going to insist a bit on something which is not that important and which is, well, a little bit annoying, but something that you have to know. And I insisted that this, so what we did, for both spectra, we calculated using these algorithms that are free for use online. We calculated the secondary structural content of both proteins, okay? There was nothing really to learn out of it, but when you have the data, this is something that you normally do. And these are the figures, and the percentage of alpha helices for both proteins is extremely low. 4% for this one, 6% for this one. If you remember what I told, you can tell immediately that there is something wrong, because I told you, this is, if you do CD, a good trick to remember. The signal at 222 is exclusively due, or almost exclusively due, to alpha helices. And for a polypeptide chain that would be 100% alpha helical, you expect a value about -30, -35,000. So you see this is inconsistent with this very low percentage. And actually, we know the X-ray structure of both proteins. We've solved that as well. I will show you in a minute. For this one, we calculated from our structure 15% alpha helices versus 4. For this one, 18 versus 6. Okay? So there is really something inconsistent there. And much less accurate, but to have a rough idea of the alpha helical content, if you measure it just at 222, for BMP2 we computed 18%, which is much better, and for this one, 23%. So, I mean, there are a number of explanations, but it's just if you need to reply something to an annoying reviewer, okay, we could say that these proteins, when they are bound to each other, change their conformation, or we could say that the proteins, when they are in solution or crystallized, display a different conformations. I think this is all, I wouldn't say rubbish, but this is not the explanation. Probably, especially for this protein, you see it displays such a weird far UVCD spectrum that there is no equivalent in the data bank. So that's one explanation. And another explanation, I've told you there are plenty of disulfide bridges in these proteins, and the contribution of the disulfide bridges in the far UV is not very well described. So anyway, this was, it doesn't deserve such a long discussion, but this is good that you realize that when you do this sort of analysis, you have to take it very carefully. So in any case, this data indicate that correct is a bit too much, but that those proteins display well-organized secondary and tertiary structure. Okay, this is the right time for me to show you the guy who did most of the work, Charlie Robert. So he was a PhD student with me, graduated last September. And this picture was taken when Charlie went to the Soleil cyclotron nearby Paris. And he obtained, in this case, this spectrum at the cyclotron. Okay. Let's move further. So Charlie measured also the interaction between the two proteins. So he measured the kinetics of binding and unbinding. For this, he used biolayer interferometry, which I briefly mentioned during the presentation of the formulation screen. So you see these methods, you can, in terms of the information it gives you, you can compare it with surface plasmon resonance, but the optical principle is not the same. It doesn't really matter. So we use these biosensors. In this case, we used biosensors that were coated with streptavidin. So upon biotinylation of Noggin, which is this considered here as the ligand, we could immobilize Noggin onto the biosensors, and by plunging this biosensor into a solution containing the BMP2, which is then the analyte, we could follow the kinetics of association and dissociation. You just see here kinetics obtained at different concentration of BMP2. And so from this data, we use a simple 1-1 binding model to fit to the data. You see that the fit is reasonably satisfactory. And we could compute the corresponding binding parameters. So the association constant, you see this is 1.2 times 10 to the 6, molar minus 1, second minus 1. So it's quite high. And as far as the dissociation constant is concerned, it's, on the contrary, quite low, 4 times 10 to the minus 4 per second. And from the ratio, you can compute an affinity, or a binding constant, if you want to, which is actually a dissociation constant of 0.4 nanomolar, which fits very well, which is in good agreement with data obtained by others using a surface plasmon resonance. So this is just another good indication that both proteins corresponds to our expectation. Yes, and this was measured at the Robotin platform in Liège that I briefly mentioned, that Maxi mentioned as well. And since last October, we have finally a manager with a permanent position. That's Marilène van der Ven, who helped Charlie to get this data. Last but not least, before the structure, Charlie performed real biological activity tests. So for this, he used a very classical model, cellular model for chondrogenesis. These are called RTDC5 cells. It doesn't really matter. And he monitored the differentiation, so chondrogenesis, using 3 complementary in vitro cell-based assays, which I'm going to describe briefly. So in a first assay, Charlie measured the activity of an enzyme. This is alkaline phosphatase, and this enzyme is overproduced during the differentiation of mature chondrocytes into hypertrophic chondrocytes. In other words, when the cartilage is, or the cartilage matrix is mineralized and transformed into bones, this enzyme is overproduced, and you can measure its activity. In a second essay, Charlie used real-time qualitative PCR techniques to measure the expression or overexpression of two genes, COL2A1 and COL10A1. These genes correspond to the expression of collagen, which is typical of cartilage, and collagen, which is typical of bones. And finally, as a third technique, Charlie used collagen staining. He used two dyes, one which is typical for cartilage formation, and another one which is typical, for cartilage degradation and mineralization. This is the brief description of the procedure that Charlie followed. So cells were cultured in a very classical way, possibly in the presence of BMP2, with or without Noggin, okay? And then the cells were allowed to differentiate for 1, 2, or 4 weeks before the characterization was performed using these three tests. Let me show you the data. Alkaline phosphatase activity assays. As you can see, when BMP2 was added at increasing concentration, this is the left-hand side panel, so activity of the enzymes versus concentration of BMP2. No BMP2, there is a very low basal activity. 2 nanomolar, no effect, but when 10 or even better 20 nanomolar of BMP2 was added, you see that there was a very significant increase of the enzymatic activity. And this was measured after 7 days of cell differentiation. Same experiment, but now in the presence of Noggin. You see that when Noggin was present in the medium, there was a very clear inhibitory effect. Okay? Second cellular assays, real-time qPCR to measure the expression of these two genes. This was measured after 1 week and 2 weeks. The most spectacular is after 2 weeks, but there is also some effect after 1 week. But let's look at the data after 2 weeks. In the absence of BMP2, no expression of this gene, no expression of that one. In the presence of 20 nanomolar BMP2, you see that there is already significant expression after 1 week, even more after 2 weeks. And finally, in the presence of Noggin, inhibition, I would say total inhibition in this case, not total, but significant in this case. And finally, the third assay using these dyes. So Charlie used Alcian blue, which is a dye specific or at least characteristic, or which binds to the glycosaminoglycans that are produced during cartilage synthesis. So you see that, and this was measured especially after 2 weeks, you see that in the presence of 20 nanomolar BMP2, there is a very significant increase in Alcian blue binding, which is indeed inhibited in the presence of equimolar concentration of Noggin. And as far as mineralization is concerned, so bone formation, Charlie used another dye, alizarin red, which binds to calcium, which is typical of bone formation. So you see after 2 weeks, no BMP added, BMP, so a massive increase in the coloration, which is virtually totally inhibited, or totally inhibited actually, in the presence of an equimolar of Noggin, concentration of Noggin. So with this, I've shown you that using these quality control assays, following the guidelines that we discussed this morning, we could show that the two proteins were pure, homogeneous, had the right identity and integrity, and probably even much more significant, both proteins displayed the right biological activity. So everything seems to be under control. We can proceed further. If you want to interrupt me and to ask questions during the presentation, I don't mind. Otherwise, we keep it for afterward. So since everything seemed to be fine, we went for for crystallization assays which worked, and we could solve the structure of the dimer. There is no big surprise because it looks very similar to the complex that was previously published between Noggin and BMP7. But okay, we were interested in BMP2, so the binding interface is not exactly the same, and so it was useful to get this structure. We obtained it at 3.1 resolution, and clearly you see that one dimer of Noggin, here in red, binds a bit. It's really like a sort of a clamp bind to one dimer of BMP2. This was done also in Liège in collaboration with Frédéric Kierf, who is our structural biologist at the CIP. Okay, this is what I've just told you. The overall structure reveals that indeed the antagonist completely masks both the type 1 and type 2 receptor binding sites of the cytokine, and also the interaction with Noggin, similarly to the other known antagonists, gremlin 1, gremlin 2, for instance, simply inhibits, precludes binding to the receptors. And so it prevents the signaling cascade. Further analysis of the overall structures reveals that the binding site between the antagonist and the cytokine can be divided, if you want to, into 3 different contact regions that are framed here. So we looked at that into more details. And on this basis, we design and we characterize so far 4 variants of BMP2, which from, I've written should, but which we hope would have a reduced affinity for the antagonist while retaining their affinity for the membrane receptors. So at this level, I should say, and I really had to do this, but I cannot give you any details about these mutants, apart for this one, which I'm going to comment a little bit. I cannot tell you into details about these mutants because this has not been published yet, and it may be patented because it has some clinical potential. Thank you very much. But this one, I mean, this is, let's say, in press, so I can explain at least this mutant. So we are in this area of the contact region, and there we identified an asparagine side chains, which, as you can read, contributes, and you can see it, not the side chain, but the residue, it contributes 4 out of the 12 hydrogen bonds formed between the 2 proteins, plus, and this is not obvious on this picture, but these side chains apparently facilitate the insertion of Noggin into the type 1 receptor binding sites. So on this basis, we expected this mutation to lead to the loss of at least 2 H bonds and to introduce steric repulsion. This is just a model, so we expected quite a significant effect. But it didn't work as we expected, since the comparison of the wild type and of the mutant BMP2, and in particular their interaction with the antagonist, showed just no significant difference. So from this, the way we interpreted these results is that the Noggin molecule is quite flexible and it has sufficient dynamics to adjust to the mutation. And this flexibility or dynamics is, I would say, obvious from the X-ray data and that I'm not going to detail, but which indicate that indeed Noggin is quite flexible. So this is an explanation for the absence of effect at this stage. So let me conclude at this stage. So I've shown you we produce large quantities of pure, homogeneous, and most importantly, biologically active Noggin and BMP2. We developed robust assays to measure the biological activity of the two proteins in the context of bone and cartilage formation, which is what we are interested in. We collected all the structural data, at least we think, that are necessary for the design of BMP2 mutant, which would display reduced affinity for the antagonist, but not for the type 1 and type 2 receptors. And we have engaged in the production and characterization of this mutant towards molecules called biobetters. That means we hope to get mutants of BMP2 which would work better than the original molecule. And finally, the data that I've just briefly commented concerning this aspartate mutant suggests that the flexibility of the antagonist is a parameter to be considered for the design of BMP2 variants with reduced affinity for the antagonist. Okay, finally, as usual, you want to acknowledge the people who really did the work, since I did nothing, at least technically or experimentally. So I mentioned Charlie already, who did most of it. Fred and Marilène I mentioned. Fabrice Bouyen, who unfortunately has left the group after more than 20 years of service. We were complaining at lunchtime about the difficulty to have, you call that research engineers. This guy was with us for 25 years or so at the protein production facility. We could never get a permanent position for him. There was a call within the university for a really, I would say, uninteresting job, but this guy needed stability. He went for it, and I understand that. Okay, and this work was a collaboration with, let's say it's a research institute called the CER Group, and this guy is one of the leaders there, Patrice Filet, and he's actually the person who came to me with the idea of this project of using and developing biobetters from BMPs. This guy at the CER did the mass spectrometry analysis, and Franz Brugg helped Charlie to develop the biological assays. And finally, funding. Again, it was quite difficult for some reason to get this project funded, but finally, we got funding for 5 years, which is quite good, from the Public Service of Wallonia, so the region where we live, which supported this project called Auto BMP2. Auto because we use the automatic workstations at this high-throughput Robotein facility. Muchas gracias.
So thank you very much, André.
You're very welcome.
Conference. So we have time, of course, for some questions. So if somebody has a question, please. Yes, student first, but okay. I consider myself students because a lot of things, especially for the protein quality. I wonder, the protein BMP is how much system you have in this protein?
In the BMP, there are 7. So 7 disulfide, well, 3 disulfide bridges in each monomer, plus an interchain disulfide, yes.
And during the, so when you express this, it's going to the inclusion body.
Yes.
And denaturation by urea, and forming by urea.
And DTT, yes.
And you arrive to 100% reformed in a good conformer?
I don't remember whether this was even measured, but the answer is for sure no.
Okay.
No. But, and actually, I'm sure, I told Charlie to try to estimate the percentage of refolding, but this is not that easy to do.
Yeah, I guess.
But, I mean, we obtain such large amounts of protein under the form of inclusion bodies that even if the refolding yield is 20%, I have in mind it's around 20%. But it's not worth the effort, to improve this yield because we had plenty. But no, it's far from 100%. Far from that.
I have another question.
So you have an anti-protomer disulfide bridge.
Yes.
And if you, I don't know if you already did these experiments or not. What happens if you remove this disulfide bridge? I mean, by mutation on the cysteine, by alanine, or by serine, whatever. If you keep the secondary structures or not?
It's an excellent question, because so these proteins are known to be dimeric. Apparently, the monomer is stable, but it's never found as an active molecule. But really, in real life, in the body, heterodimers exist.
Okay.
For instance, between BMP2 and BMP7. And this is not very well described in the literature, but apparently, some of these may be even more active than the homodimers. So at some stage, if we manage to restart the project with new financial support and with students, one of our goals would be to build, to engineer an heterodimer between BMP2 and BMP7, for instance.
May I ask a last question?
Yes, of course.
In fact, so what you said is very, very interesting. In fact,
It is. Yeah, it is.
Yeah, one of the protein is perhaps less stable or more flexible when isolated. So, do you think that could be some allostery between these two proteins upon binding?
I don't think so, but I've not thought at that. So it could be. I don't know. I don't know.
Thank you.
Thank you for the question. So you have another question too? There is some student has question? Okay, I will come back. No, it's because the streaming, it's for the streaming.
Okay. Thank you for the talk. My question is more general. It's about the relationship or what is known about BMP to Noggin in osteoporosis, and if BMP is used as a treatment or anything like that because of the fail in the bone formation.
Basically, I don't know. I can give you very much details, but what I can say is that there was a lot of enthusiasm initially for these proteins, where they were not patented, but approved by the FDA. But, as I explained at the beginning, they have so many different activities that, and this is not controlled, this is not well understood. When they are used as medicine, as drugs, beside the beneficial effect, they have plenty of adverse effects. And so far, this has not been controlled properly, and so they are not used anymore, or not much. And what should be done is to use them very locally. So, if you would have an implant of to restart, to promote bone or cartilage regeneration, an implant, using BMPs within these implants is a good perspective. But to administrate these proteins as drugs, I think this is not done anymore.
Okay, thank you. So, Amparo, do you have a question?
Thank you, André. Two questions. When do you, or why do you use urea, and what concentration do you use the urea in the refolding experiment? Why do you not use guanidine chlorure?
Guanidine chloride.
Cloruro de guanidino. Why do
It's
you use urea?
It's completely empirical. I don't think Charlie tried guanidine chloride. I don't know. But there is no rule. What you have to know, and this is so people would use urea in general rather than guanidine chloride. But there is no good reason for that. And personally, I would even prefer and recommend to use guanidine chloride because urea is not stable. It degrades quite fast with time, and it forms cyanide derivatives which react with protein. So if you leave a protein for several days in urea, you're going to modify the lysine side chains, and this can be an issue. But again, in this case, Charlie was careful enough not to, the solubilization of the protein in urea was quite fast, and so he could initiate refolding almost, not immediately, but within a few hours. and mass spectrometry indicates with absolutely no doubts that we have no chemical modification of the lysine.
Yeah.
But it's correct that for refolding inclusion bodies, I'm sure everyone or most people will go for urea, but it would be a better idea to go for guanylyl chloride. Yes?
Just a comment on this. In fact, people use, those that purify proteins use urea because immediately after purification they can do this stage. That you cannot do it by guanidine.
Oh, right, okay.
Okay.
That's it. And in
Okay.
the Hofmeister
Yes,
series,
right.
guanidine is the best chaotropics one.
Yes.
But okay, urea, me for example, I use also urea because it's much more convenient. You do STS page immediately.
Right, okay.
You cannot.
Okay.
I mean, it works as well with urea and with guanylyl. But you have to know that urea may chemically modify your protein. And many people ignore it. And since they don't do the quality control test, they may well be working with chemically modified protein. I'm sure this happens a lot.
Okay, thanks. And the native protein has a post-traductional modification? Native protein.
Good question as well. Yes, these proteins are normally significantly glycosylated.
Glycosylation.
Not in our case, of course, because they are produced in E. coli. But it was demonstrated that, and indeed, this is what we did as well, that the glycan side chains have no effect on the activity. So this was known already before we started, and as you've seen, it works. But normally they are quite significantly glycosylated, indeed.
You have a question?
BMB and Noggin was expressed in the same time in all the process for the formation of the hueso, bone. You show in the
Right.
when
In the cellular assays. you
yes.
mean? So at this stage, in the different.
My question is, in normal, in vivo,
Right.
the Noggin and BMP
I see what
are
you mean.
expressed in the same time. Ever, or for example here, what happened in the second
So-
step?
Noggin is expressed during this phase, okay, but not during this one. This I'm almost for sure. And BMPs, I think they are expressed all through the process. But Noggin are not expressed by the major chondrocytes, naturally, if that's your question. But obviously, when we do the experiment like this in vitro, Noggin is going to have an effect at this stage. But in bone formation, the major chondrocytes do not produce Noggin. This was not your question, or was it? Yeah, go ahead.
Tú hablas de flexibilidad entre Noggin y BMP y que la mutación disminuye la flexibilidad.
No, that's not what I'm saying. What I'm saying. Why are you laughing?
I'm impressed.
What?
No, because you really understood the question. So
Yeah, I hope so. No. So this mutation, because it's quite severe, I would say, we expected that it would have an effect, and it has no effect. So a possibility is that the Noggin molecules rearrange or slightly change conformation to adjust or to adapt to the mutation. This would mean, if this is correct, it means that the Noggin molecule is quite flexible, and the X-ray data for completely other reasons show that it's quite flexible indeed, because some part of the structure is missing. And also, when we compare our structure of Noggin with the structure of Noggin in complex with BMP7, there are a few significant differences. For instance, there is an alpha helix, which is like this in one crystal and like that in the other. So these are indications that Noggin is indeed very flexible. But you know, this is the sort of thing that you have to write in the conclusion of your papers. At this stage, it's clear that it is a flexible molecule. Now, whether this is the explanation for the absence of effect, we won't really know until we combine actually the various mutations that I have not shown you. I'm sorry about that. I mean, we have clear evidence that Noggin is quite flexible. Now, whether this is the reason for the absence of effect, maybe, but this is not completely sure. Have I replied to your question? Good.
Not in Spanish, but
No, but I mean, I'm already improving my level of understanding.
That
So
was really quite impressive.
no, but you speak so clearly. No, it was obvious. Thank you. Yes.
Just for a start, a comment on one question that has been raised regarding the biological roles of
Noggin.
No, not Noggin, BMP.
Right.
As it belongs to the superfamily of TGF,
Right.
yeah, I think side effects should be really kind of neoplastic or something like that. And
I don't know, but I know there are really a lot, and
sure
it's
that
more
it's
detrimental
really
than
pleiotropic and
yes.
I think it won't be relevant to use it in all body.
No, it has to be administered locally. This is very clear.
And the question that also rose to me is, why is it? Was it for you a strategy to express this BMP as with mutation just to reduce the reactivity with Noggin? Would it have been relevant to design some specialized antagonists for Noggin instead of having a BMP that is mutated in order not to recognize Noggin anymore?
So you mean it would be easier to?
I don't know anything about the biology of these two proteins, but maybe if the goal is to produce BMP that is not
Affected by the
affected
occurrence,
by
yes.
the Noggin, either
You would
could
trap Noggin rather than.
either you could trap Noggin, expecting that the endogenous PMP would be enough, or
Right.
either you would add more BMP in the organism,
Mm-hmm.
expecting that it will overflow the Noggin concentration to get active effects. Why such strategy, meaning both?
So to administrate higher quantities of BMP, well, maybe this will produce unwanted side effects. That's one thing. Another thing is that protein, I mean, before we started, so this protein is commercially available, but it's extremely expensive. And what we found in the literature, and this is something I did not insist at all on that, but the level of production of pure active Noggin and BMP2 actually that we achieve is really far above anything that has been reported so far. So this was not particularly anticipated, but it doesn't mean that we can produce kilograms to, So the idea of using more BMP to treat patients, I think this is not a good idea. No, the other possibility would be to target Noggin, you say. Yeah, but it means you need then to administrate BMP2 plus an inhibitor. Well, maybe it would, I don't know, but this is not indeed what we decided. But I mean, we are far from being there, but from the other mutants, we have indications that we may succeed at some stage if we find the resource to carry on with this project. And we have some hopes that by combining some of these mutations, there will be an effect. Now, whether it will be feasible, this is another story. This is another stage. But there is a lot of interest in the field, which is not my field, but in the field of regenerative medicine for this. There is still a lot of interest for this BMP. So
So we still have time for more questions. So the students,
they will ask questions at the coffee break
Yes.
if they have.
Perhaps last questions.
Please.
If the mutation, you don't see any, effect, real effect, did you look during the unfolding, you did some unfolding experiments to
No. To
Yeah.
see what?
to see if this modify the enthalpy or the entropy of the unfolding process?
No. We didn't do this. No. But what would you expect?
Uh,
You mean to unfold the complex?
yeah,
Okay.
you can at least see, you can at least see if the heat release during the dissociations change, and if it change, is based to the some flexibility, so entropic effect,
Right.
or enthalpic, because
So then you mean measuring the binding constants by ITC, is it what you
yeah,
mean? Or
ITC, you can do it by
Okay.
ITC, yeah.
so it's not unfolding.
But you can do it also by DSC.
Right.
But you- by
Okay.
DSC. Huh? It depends.
I mean, if it works well with ITC, you can compete with it, because with ITC you will have the entropy, the enthalpy, that would be perfect. But
Yeah,
no, we
but
haven't
you-
done that.
But you, if you do it by DSC, you will have the enthalpy and the entropy of the dissociation and activation energy too. That by
We should do both.
yes.
No, we haven't done that.
I have a question. So regarding this slide, because for me was
This one.
yes. Just because you started to design a mutant based on a structure that you solve at 3.1 Armstrong resolution.
Right.
That is not the best resolution. So
No, but we couldn't achieve better. I'm
no,
really
I know,
sorry.
but the point is you are defining here a 6 H bond, right? There is 4. Yes, 4 at least. Yes, 4 H bond that I'm not sure that you can really see them using that structure. So I don't know if you try, for example, before doing the mutation, trying to run some molecular dynamic simulation just to be sure that you have those H bond there.
No, we didn't do that, and I trusted my structural biologist colleagues.
Okay.
Something I didn't say is that also we decided to introduce an aspartate because in a homologous BMP, when you align the sequence, this residue is found in some of these BMPs. And the sequences were analyzed into details. Yeah, could be if you tell me that one of these H bonds comes from the imagination of the X-ray guys. Could be. It could be.
Okay, thank you. So